So has everybody who's been wanting to use the videos succeeded at connecting to it either through Icon or through Google? Is there anybody who's wanted to see a video and has failed with both of them? Is there anybody on a Mac who's succeeded at Icon? Oh, what do you use? On a Macintosh on Icon? What browser did you use? Safari? I'll have to try that. I thought I tried it, but I get sound but no picture when I use Safari on Icon, so who knows? Okay. Any questions about the discussions in Chapter 4 about nucleic acids, DNA, RNA, genetic code, things like that? Or about proteins, hemoglobin and myoglobin? That would be Chapter 4. Chapter 3 was DNA. Chapter 4, hemoglobin and myoglobin, before we go on to the cytoskeleton. I'm sure you're all aware of the fact that we're approaching our first hour exam, which will be on Friday, unless, of course, we get snowed out again. I do ask you to make every effort you can to be on time. I understand some people cannot be here at 3:30. However, to avoid disturbing people who are already working, anybody who arrives after 3:30 will be asked to wait outside and will be seated as a group at 3:40. If someone arrives later than that, we'll work on that. So please try to be on time, and if you can't arrive before 3:30, understand that we'll ask you to wait outside. The other thing I'd like you to have in mind for the test on Friday is try to bring with you your university ID or something that has your university ID number on it. We have found it really helps if we cross-check what you've coded on the sheet to your ID card. It reduces the number of errors tremendously. In the past, we would have 25 to 30 miscoded ID numbers, which exhausts everybody trying to straighten it out. So if we check your number, it comes out right. So try to bring your university ID or something that has your university ID number on it. And we'll just cross-check that as we leave. Do people have other questions about the exam coming up on Friday? It's chapters 1 through 5. Was there a question over here? Okay. Can people see with that, or should I turn it down a little further still? What are people's thoughts? Let's see, is that where we are? Okay. If people need it darker, let me know. There's always this trade-off between awake and seeing things. The next chapter, Chapter 5, is going to address a surprisingly large fraction of the cell. It addresses a structure that's only come to be known really in the last 25 to 30 years. But the cytoskeleton can comprise anywhere from 15 to 90% of the cell mass. So as a structure made out of proteins, it is one that's extremely important. Some of the things that the cytoskeleton do are to provide shapes, and the shape of a cell will determine how it can function. So for instance, a red cell that wasn't round doesn't work. A muscle that's not long and skinny doesn't work. The cytoskeleton is the basis for movement, and movement of many different kinds. So it's for movement involved in cell division, movement of actually swimming through liquids, as algae can do. Not very many of our body cells do it. And although we make liquid move, and that's a separate question, or cells moving by crawling, which is an important part of normal healing. It's an important part of metastasis in cancer. The cytoskeleton is also very important as the basis for mechanical strength, that we hold together as an organism in large part because of the cytoskeleton and extracellular matrix. The cytoskeleton is also one of the most important markers for differentiation. That is, to know what cell you're looking at is often to read its cytoskeleton. When we look at a cell and say, that's a neuron, the things we see in the microscope, the shape and the processes and so forth, are largely a matter of the organization of the cytoskeleton. The biochemistry comes second. And finally, the cytoskeleton is intimately involved in cancer because it's one of the hallmarks of a cell that's on the pathway to cancer, and it's part of the way in which cells uncouple themselves from normal regulation, that the cytoskeleton is usually actively transducing important control information, and in cancer that transduction fails. This little thumbnail picture here, and I'm going to turn the lights down a little more, I think. I think that's further down. Is that further down? Oh, dang, I didn't do it. Can someone take those spots off again? Oh, well. I guess you can see through them. What this panel of pictures is, can you see to take notes, or am I going to have to go back up? Okay. What this panel of 3 pictures is the same cell in which 3 different filament systems have been revealed. And so the top picture is showing you the actin fibers. These are bundles of actin microfilaments. This is showing you in blue microtubules, which we're going to see are especially addressing compression bearing. And here are intermediate filaments. And one of the things that I wanted to come across to you is that these 3 filament systems are not completely independent of each other, but they are interacting with each other in order to accomplish their mutually shared tasks. So let's look at the first group of filaments called the microfilaments. The microfilaments are called that because they're the thinnest of the cytoskeletal filaments. They're about 7 nanometers across. They're extremely rich underneath cell membranes, where you often can find them as a thick kind of a felt, especially in cells that are actively crawling. Actin microfilaments are often found in bundles or in cables, so that although one microfilament's not terribly strong by itself, you put a bunch of them together and they are. Oops, I'm sorry, that should be, there we go. And what's interesting is that although the actin microfilaments are long, skinny filaments, what they're built up out of are actually globular proteins. And this is a picture here, a ribbon diagram of an actin monomer. And this is the subunit of microfilaments. And if you have enough of these actin subunits together, you can make a filament. And if you look at this carefully, you'll see that it's actually 2 helices twined together, what we call a 2-start helix. This helix is polar in the sense that one end is different from the other end. And we're going to see why one end is different from the other end in just a second. The actin monomer can bind nucleotides, and what it binds specifically is either ATP or ADP. And we're going to see that which it binds will make a difference to how it's going to behave, but it's one or the other of those 2. We call it F-actin when it's in the filament for filamentous actin, and we call this G-actin because it's the globular monomer. And this is an electron micrograph of an actual microfilament, and this is a reconstruction of what the texture of that filament would look like. Now, actin can be polymerized, but this is not a covalent polymerization. When we talked about using nucleotides to make a polymer of nucleic acids, there's a covalent reaction that goes on to make a covalently bonded, long, skinny thing. When actin polymerizes, it's not going to actually become cross-linked. It's just going to associate in a reversible way. And if you look at an actin filament, you can see here that it's a helical, it's a twisted structure, and it's a polar structure in this respect, that the 2 ends of it behave differently. And we often call those 2 ends plus and the minus end, or the slow and the fast end. And what that is pointing towards is the fact that the minus end, or the slow end, binds the globular actin fairly slowly, and the plus end binds it more avidly or more quickly, so that this end will grow more slowly, and this end will grow more quickly. The earliest step of polymerization is fairly slow. If you have a solution of completely globular actin with all G-actin, to get from that to any kind of a filament is a fairly slow process, because you have to get 2 to 3 subunits to stick together long enough to be a nucleus for the next thing to happen. But once that happens, once nucleation has happened, the elongation part is quite quick. It's especially quick off the fast end. It will still happen, but not as quickly off the slow end. Now, what's interesting is as this growth is occurring, both at the fast end and the slow end, or the plus end and the minus end, the ATP that was on that G-actin is hydrolyzed to ADP. So if you're looking at an ATP subunit, it's generally going to have an actin subunit, globular actin subunit. It'll usually have ATP bound. If you're looking inside the filament, it's usually going to be the hydrolyzed ADP. Now, the fact that the 2 ends are different, and the fact that there's hydrolysis of ATP involved means that the dynamics of all this can get somewhat complicated. If you imagine this polymerization process going and going, at some point you're going to have so much polymer and so little monomer that the rate at which things come off is going to equal the rate at which they go on. There'll be a dynamic state that's reached. And at that state, you have new subunits being added to the plus end at the same rate you're losing them from the minus end. And in the test tube, that can be shown to result in something that's called treadmilling, where if you wanted to track this particular pair of actin monomers, you can see that it's in the middle in that first diagram, but as addition occurs at the plus end and loss occurs at the minus end, it's coming closer and closer to the end. In other words, it's like it's treadmilling along through that thing until it comes out the other end. For true treadmilling to occur, you have to have both ends free. In the cell, in fact, both ends are not usually free. But the treadmilling we observe in the test tube does point to the fact that the filament is very definitely polar and that the ends behave in very different ways. Another way in which we can see that the actin filament is polar or asymmetric is that there are proteins that interact with it in very specific ways at different parts of the filament to do different things. So, for example, there are proteins that can bind to the plus end or the minus end to cap that filament to stop further addition or loss. There are proteins that can bind on the side and branch that. So instead of having a spaghetti pile, you now have a web of actin filaments. There are depolymerizing proteins that can accelerate the rate of breakdown. There are some that can simply split it into two. And all of these different accessory proteins can then modify the rate at which actin filaments form or break down. This is giving you a little example of how if you were to remove the capping protein, you could have that uncapped end now be available for growth or available for breakdown, depending on what it's doing. We know that these actin dynamics are important because it's been shown by a number of different methods that for a cell to actually crawl actively, where it's sending out a cell membrane rich in actin filaments, for it to actively crawl needs new polymerization onto those actin filaments. So these dynamics are not just a test tube phenomenon. They are an important part of actually living, moving, crawling cells. This is just to give you a little bit of a feeling for some of the ways in which actin-based movement makes a difference. So actin polymerization is an important part of crawling in most cells that crawl. And you can see here, this is an electron micrograph to show you how very dense the actin filaments can be at that edge of the cell that's crawling actively. This is a scanning electron micrograph to show you the overall shape of a cell. This is the front end or the leading end, and that membrane is just ruffling like a window curtain. And this is the tail that is attached and at some point will let go and snap forward. We see extensive cell crawling during embryogenesis, so that sorting out which cells go where, a part that happens through crawling. We see it during wound healing, that either when you cut yourself, when that has to heal, or in artificial wounds in vitro, cells crawl to fill in that space. And finally, cancers that are strongly metastatic, that are very good at establishing colonies elsewhere in the body, in most cases are also very good at crawling, that they can crawl through the extracellular matrix, they can crawl into tissues and wreak a lot of damage that way. Now, in addition to actin polymerization, other things are needed for actin filaments to provide, or microfilaments to provide, the full range of cell movement. And one important component of that is the molecule myosin. Myosin is what we call a motor protein. So it's a protein, but it's a protein that uses chemical energy to make things move, which is pretty cool when you think about it. And when we look at the structure of myosin, we can see how the structure of myosin might enable it to perform that function. What we see when we see myosin molecule, this is an electron micrograph here, so you can kind of trace this long tail and then these two heads. This is a diagram to show you that in a little more detail. So you actually have two peptides here that are twined around each other with globular heads at one end of the molecule. In this area, there generally is additional polypeptides called light chains that are much smaller than the myosin heavy chains. And these heads are capable of hydrolyzing ATP to go through shape changes. And ATP hydrolysis is coupled to these heads walking along an actin filament. This long tail portion of the molecule in some myosin molecules allows it to make a bundle like this. You can see there's a thick core here in the center, and then the little head groups are sticking off of it. This would be called a thick filament. It's quite a bit thicker than a microfilament or thin filament, and it's in fact the thickest of the filaments that we'll be talking about. This is looking in a little more detail at the how it is that myosin and actin can interact. This is showing you a more detailed picture of the structure of a myosin head and its neck. And the kind of detail that we have now is such that we can identify both a place for ATP to bind and a place for ATP actin to bind. So the head portion of myosin is structurally distinct. This is showing you at a higher level of organization. Now, this is about a micron from there to there. Here is a thick filament with many myosin molecules in it. Here's a thin filament with many actin molecules in it. And what happens is these myosin heads walk along that thin filament. And as they walk along, they're walking in opposite directions. They're pulling that thin filament past itself. And so you can see here that the myosin thick filament is the same length that it was before, but it has pulled the actin filaments in closer to the middle of it. In fact, right up to the end of the heads, because now there's nothing else to pull on those actin filaments. So the contraction of muscles is accomplished without anything actually itself shrinking. The actin filaments are the same length, the myosin filaments are the same length, but they have slid past each other to make the muscle as a whole contract. Any questions about that? Myosin crawling on actin is an example of what's called mechanochemistry. The idea that you can have mechanical machines driven by chemical reactions at the level of cells. And it's just one of a number of examples. We're not going to go through all the ones that do it, but I want to sort of introduce you to this one because it's the first one that was understood and one of great importance. Let's just start here in, it's a cycle, so we could start anywhere, but let's just start at this position in which the myosin head is bound tightly to an actin filament. If an ATP molecule comes in and binds to the myosin, that will allow the myosin head to come off from the actin filament at that point. When the ATP that's in the myosin head hydrolyzes and becomes ADP and an inorganic phosphate, that causes a change in shape in this region, so it flips the head forward. The myosin head group can now bind again to actin with, at this point, still the ADP and PI bound. And when it releases that hydrolyzed nucleotide, it flexes backwards, and that's the pulling stroke, the power stroke. Okay. So it's kind of an interesting thing that the hydrolysis step is separated a couple of steps from the actual movement that generates movement by these things. But it's an ongoing cycle that has to occur more than once to end up with a net muscle contraction. This kind of motion by myosin of using ATP hydrolysis to pull itself along an actin filament or to pull an actin filament past itself is obviously important in our muscles moving our bodies. It's also important in cell division. It's part of how cells pinch off to make two cells. It's an important part of cell movement. Although actin polymerization is part of it, myosin contraction is another part of it. I've already mentioned muscle contraction. And there are other forms of myosin, a little different from the one I just showed you, but still the same general biochemistry, that are involved with moving particles within the cell. So it's a very diverse molecule with a finger in a lot of pies. And I think I'll come back to these if we have time. I see it's already 10 to 4. So I'll come back to these. If we don't have time to do these today in class together, please take a moment to go to them on your own, because it shows you something rather neat, which is now we can label these cytoskeletal proteins with green fluorescent protein and actually see them do things in real time. And we don't have to reconstruct the cell behavior out of several different pictures. We can watch it in real time. So if we have time, I'll come back to that, but I want to make sure we get to the other things first. What happened? There we go. The second major class of cytoskeletal structures are called microtubules. We're going to see that they're called tubules because they actually seem to have a little hollow in the middle. And because they're tubules, if you think about like the empty cardboard tube in the center of a toilet roll paper or wrapping paper thing, that tube is remarkably strong if you push down on it. You can actually build little chairs and stuff out of them. And so microtubules are very good at making things rigid or bearing compression, pushing in like that. The microfilaments are good under tension. Microtubules are good in compression. Microtubules are built out of dimers, not out of monomers. The dimers are an alpha-beta dimer of tubulin, and they can bind GTP and GDP. The alpha-tubulin only has GTP bound, and it has that GTP bound all the time. That's just what it has. It doesn't vary. The beta-tubulin can bind GTP, and then it will hydrolyze that GTP after polymerization, just as we saw actin doing with ATP. And what this means is that the microtubule, just like the microfilament, is a polar structure. Its ends are different. And we'll take a look at how that is in a second. So here's sort of a diagram of an overall microtubule that's in the process of growing. And you can see here that it actually is a tubule. It's got a number of subunits organized around in a cylinder. And we can see that that the tubulin dimers can associate in a number of ways. They can make a protofilament in which the alpha-beta subunits are head to tail. These protofilaments can associate side to side. And then if enough of those have associated side to side, they will close into that tubule itself. And this kind of association from filaments to tubules is more common in test tubes. When we look in actual cells, there are pre-existing circular structures that are probably the most commonplace for nucleation for microtubules to happen. But microtubules, like microfilaments, need some kind of a nucleus to form off of. And they have a plus end and a minus end. That is, they have an end at which addition is faster and another end at which addition is slower. You can also see from the way this diagram is drawn that there's a polarity to the organization of it, that the alpha and beta subunits are lined up in one particular way all the way up the microtubule. Just as actin filaments came apart at one end as well as assembling, microtubules can depolymerize. One way that you can see this is that if you take a solution of microtubules and dilute it, so there's many fewer tubulin dimers available for it, you may see the ends of the microtubules fraying like this because they're starting to peel apart. In contrast, in a cell, generally speaking, the minus end, the slower growing end of the microtubule is often anchored on something. And it would be the positive end that would be the most actively changing end. But it would also tend to have proteins associated with it, just as microfilaments. That it's rarely a free end in the sense of just being naked in space or something. And just as we saw for microfilaments, there are a variety of filament-associated proteins in this case. There are ones that can bundle microtubules. There are ones that can cross-link microtubules into kind of a felt and so on. Because microtubules are so intimately involved with mitosis, different kind of mitosis than the slides before class, microtubules are required for the separation of chromosomes during cell division. Now, cancer cells in general are undergoing growth at an unregulated rate. They're having multiple rounds of mitosis that they shouldn't be having, or beyond what they should be having. And there are antimicrotubial drugs, two of which are vinblastine or Taxol, which, because they interfere with the correct balance of microtubules, prevent that mitosis from occurring in a very good way. So they make good anti-cancer drugs in that they target the cancer cells more than the rest of the body. It's not an either-or. They will affect the rest of the cells in the body, but the rest of the cells in the body aren't going through mitosis as often, and so they're not as affected as much. This is showing you a picture of some of those structures. You can sort of get a hunch that possibly some of these have affinities to the shape of GTP, but it's not perfectly clear what all they come from. Taxol is an unusual drug because for many years it could only be isolated from U. It could not be made chemically. Recently that synthesis has been accomplished. So we talked about microfilaments being made up of actin, being polar, and having motor proteins, especially myosin. Well, it turns out that microtubules also have motors associated with them, but different ones. And in the case of the actin filaments, the filaments slid past each other. In the case of microtubules, the motor proteins will usually move things along the microtubules. And a couple of the things that are especially conspicuous there are the chromosomes. So here you have a rather beautiful picture of a dividing cell. The blue here are the chromosomes themselves, looking a little bit like sausages. And then the green here are the microtubules with the red spot at the centrosome. And these chromosomes are going to move towards that centrosome, partly through the action of a microtubule-based motor. And as you can imagine from what we saw about myosin, microtubule motors will have a particular direction that they're moving on the microfilament. This particular one is a picture of a protein called kinesin. It's another motor protein, specific in this case to microtubules. It also has a long tail. It also has two heads. And in the case of kinesin, the light chains are not near the head groups, the way it is in myosin. The light chains for kinesin are down at the far end, and these light chains are involved in attaching the kinesin to cargo of various kinds, vesicles that the cell is trying to move to somewhere else in the cell. This is to give you a little bit of an overview of how the kinesin is going to help out with all of that. You can see that in a picture from the earlier page, rather larger here. Here you can actually see the structure of the kinesin at the level of a ribbon diagram and how little there is really at the neck between the head groups and the tail here. Here they're trying to draw more or less to scale, although actually the cargo could be larger still, showing you how these light chains have attached a vesicle of cargo onto the kinesin, and the kinesin is moving that cargo towards the plus end of the microtubule. In general, the minus ends of the microtubule will be near the centrosome, near the center of the cell. The plus ends will be out at the periphery of the cell or near the chromosomes. So this is moving it either out towards the periphery of the cell or towards a chromosome. Kinesin is a little different from myosin in terms of how it accomplishes that. The myosin had kind of a hinge-like neck. Kinesin is a little more floppy. And so in the case of kinesin, when there's no ATP bound, it can be bound to the microtubule. But if it does bind an ATP molecule, so here's an ATP molecule binding, it can change its shape, and it sort of flings the trailing head forward. So it's kind of twitching or pitching. And you can see here the head that had been behind being pitched forward. And when the head that was behind and had an ATP bound to it binds to the microtubule, it will release the ADP. And now you have the trailing one still has an ATP on it. The leading one is empty. When the trailing one hydrolyzes its ATP into inorganic phosphate, it can now lift up off the microtubule and be ready to go. So one of the interesting things here is that there's always one foot on the microtubule. It never lets go completely. And so we call the motion of kinesin processive. We're going to run across a number of things in biochemistry that are processive, but they're enzymes that can do something over and over again to a long, skinny substrate. In this case, the long, skinny substrate is the microtubule, and the kinesin can do hundreds and even thousands of these reactions, pulling its cargo out. If you think of how long a neuron is, you can see that kinesin has to be able to keep going a long time to get those vesicles out to that end of that neuron. Okay. Any questions about microfilaments or microtubules before we start on intermediate filaments? Okay. Well, the third family of cytoskeletal proteins, big family, are the intermediate filaments. They're called intermediate because they're bigger than microfilaments made out of actin. They're smaller than thick filaments made out of myosin. That's why they got their name. And they are far more diverse than either the microfilaments or the microtubules. There are at least 5 major families. Within each of those families, there are several to dozens of members. So there's a lot more kinds of intermediate filaments than there are kinds of microtubules or microfilaments. In the epithelia, like skin or the lining of the mouth, there's a family called the keratins. Keratins are especially conspicuous in tissues that are very much subject to mechanical stress. And that's a very common role for intermediate filaments to play, is a structural role resisting stress of various kinds, mechanical stress of various kinds. Now, we're going to see from the way the filaments are made that they are not polar. Microfilaments and microtubules are. They have these nucleotides bound and plus and minus ends. Not going to be true for intermediate filaments. They are going to be symmetrical whichever way you look at them. They also don't bind any nucleotides. They're forming coiled coils, and this will be one of the first times that we'll talk about a very common protein motif, and I'll show that to you in a little more detail. However, we do see interactions between intermediate filaments and microtubules, between intermediate filaments and microfilaments. So if you want to think about it, you can think about microtubules as kind of the bones of the cell, the microfilaments as the muscle of the cell, intermediate filaments as the tendons and sinews of the cell. Now, a large fraction of intermediate filament protein is taken up in what are called coiled coils. And this is a very, very common structural motif. It's a coiled coil because it's taking 2 polypeptide chains and coiling them around each other. So there's one alpha helix here, another alpha helix here, and the 2 alpha helixes then are coiled around each other. So the first coil is an alpha helix. You take 2 alpha helices and coil them, you now have a coiled coil. It's the predominant form of interaction in the intermediate filaments, but it's common in many settings. So for instance, the tails of both myosin and kinesin are coiled coils. We're going to see that transcription factors often have coiled coils. It's a motif that shows up in many contexts, not just structural proteins. Now, the reason that a coiled coil works is because when you look down that alpha helix, you can see that at the 1 and 4 positions, looking down the repeating coil of the alpha helix, the 1 and 4 positions tend to be hydrophobic. And so if you have hydrophobic things at the 1 and 4 positions on this side and on this side, when they coil around each other, they tend to touch each other. And so all up and down this contact between the 2 alpha helices is like a ribbon of hydrophobic contact. Now what's interesting is the coiled coil itself is a left-handed helix. So we know that the alpha helices themselves are right-handed, but here you have a picture of a coiled coil. So the blue is one alpha helix, the green is another alpha helix, but when they twist around each other to make a coiled coil, that's a left-handed helix, a 2-start left-handed helix. Any questions about that? So if we want to think about how intermediate filaments assemble, we can look at the parts of it being made. We can look at the parts that go into making one. The monomer here is a single polypeptide. 2 of those together can make a coiled coil or a dimer. If we pack more of those together, the dimers can be staggered relative to other dimers, and that would make then a tetramer with ragged ends, if you like. So it's a filament of staggered dimers. Taking several of these together, we can make an octamer, and then bundled together and so forth. So you're making a progressively more bundled set of filaments with each other. But if you notice the way at which the tetramer, if you look at the way that the coiled dimer is, it has a polarity to it. It has its N terminus and C terminus, aligned. It's a parallel coiled coil. But if you look inside the tetramer, that polarity is lost. So overall, there's no polarity to the filament as a whole, number one. Number two, they're very insoluble. All of the intermediate filaments are insoluble to a considerable degree. Some of them are horribly insoluble. There are no nucleotides bound, and so there's no nucleotide hydrolysis. It's been shown that for one of the intermediate filaments called vimentin, some of that vimentin actually starts making filaments as it's being translated, which might make sense when you think of how very insoluble these proteins are. And as you might think, for something that's assembling during translation, its messenger RNA is also organized into a location that's good for where they want the filaments to be. Okay, any questions about those three filament classes, the microfilaments, the microtubules, the intermediate filaments, the three major filament systems within the cell? Any other questions about them? Okay. Let's look at another structural protein. This is collagen. It's not per se a part of the cytoskeleton, which is inside the cell, but it is a structural protein, and I think that's why they included it here. Collagen is a very abundant protein. It's outside of our cells. It's a major part of skin. It's a major part of tendons and so on. And it's a very funny protein in terms of its composition. You probably remember that proline has this odd structure where it's a closed ring that closes around on to the N nitrogen of the amino end of the amino acid. So you have a kind of a constrained structure to it, that the proline is not free to twist the way some of the other amino acids would be free. So this is proline. The other amino acid that collagen has a lot of is hydroxyproline, and you can see here a hydroxyl group that's been added. That hydroxyl group is added after the peptide is synthesized, and it requires vitamin C. And it's because collagen needs to have that hydroxyl group put on it that absence of vitamin C gives you scurvy. Your connective tissues slowly fall apart. So this is kind of an interesting twist. The collagen polypeptide is also an extended coil, but it has a form that is pretty much unique to collagen. I'm not familiar with any other of the proteins that takes on this tertiary, this secondary or quaternary structure. It has an extended left-handed helix. The coil of a collagen molecule is a more extended left-handed helix than a typical alpha helix. And three of these together will make a triple coil like this. And then it has a right-handed turn to it. So three collagen molecules together make a triple helix that has a right-handed turn around to it. Each one of these triple coils can then associate with many other of them, and you can end up with quite substantial bundles of collagen, as you can see here. This is an electron micrograph showing you so many lined up side by side. You can actually see stripes to the organization. Just as we saw a repeating motif in the keratins of the first and four positions being hydrophobic, and that gave it the capacity to make a coiled coil, the sequence to collagen also has a repeating motif, and it's an extremely simple-minded motif. It's glycine, proline, hydroxyproline. Now, there are other amino acids in collagen, but that accounts for a big chunk of collagen. And by repeating that, again, glycine, proline, hydroxyproline many times, it makes possible this extended helix, this elongated left-handed helix. And when you look inside the triple helix of collagen, in that central part of it, it is so tightly packed together that the only thing that there's room for is glycine. And so that accounts for why the glycine is in that third position all the time. It also accounts for why chicken soup is slightly sweet. Glycine is one of the common amino acids in chicken broth, and it gets its name from the fact that it tastes sweet. Just so you know that biochemistry is out there in daily life. If all that our bodies had was collagen that was formed from these non-covalent interactions with the glycines in the center of the triple helix, we would not really be strong enough to hold together. There's additional chemistry that goes on after the assembly of those fibrils of collagen, in which at occasional points along the molecule there are lysine residues. They're not as abundant as the proline and hydroxyproline, but they're in there. And where the lysine molecules are in the correct position, they can actually become covalently cross-linked to each other. So the collagen fibers become, in effect, one huge molecule because they've been covalently cross-linked to each other. Sorry, yeah? Oh, that's not on your handout? Huh, let me just see. So it's not, okay. I'm sorry. Let me just take a quick look at, okay, that was the last of it, so that's okay. Let me back up, and it means we do have time to go look at the movies, which I think are really worthwhile. Here we go. So I'm backing up to slide number 9 here. Again, the idea here is that the green fluorescent protein is going to highlight for you what the protein is doing in the living cell. So if all goes well, here we have a fibroblast moving with GFP actin. Oh, come on. Okay. What's the problem here? Or maybe we don't. I know this worked on my browser. Maybe we finally killed the browser. Okay, just let me remind you that there are office hours available for you tomorrow. Both of you are having them, correct? Both Tim and Lynn will have hours tomorrow, which if you have questions coming up for the test. So make sure you keep that in mind.